Welcome back, my dear students. Today we will explore an important topic in carbohydrate biochemistry, that is disaccharides and polysaccharides. These are carbohydrates formed by the combination of simple sugars or monosaccharides through glycosidic bonds. Disaccharides are basically such as maltose, sucrose and lactose are made up of two monosaccharide units joined by O-glycosidic linkage formed through a dehydration reaction. They serve as an important sources of energy and act as intermediates in metabolic processes. Polysaccharides, on the other hand, are complex carbohydrates formed by the polymerization of many monosaccharide units. Examples include starch and glycan, which are the energy storage polysaccharides, and cellulose and chitin, which are the structural polysaccharides. Their structure can be linear or branched depending on the type of linkage present, whether it is alpha or beta glycosidic bonds. These macromolecules play vital roles in energy storage, structural support and biological recognition in living organisms. Glycosidic bond and formation of disaccharide. Now we have to know what is a glycosidic bond. Basically, a glycosidic bond or linkage is a covalent bond which is formed between two monosaccharide units. It forms when the hemiacetal OH group of one sugar reacts with the OH group of another sugar with a loss of the water molecule. This is the loss of water molecule or condensation reaction, and the bond forms between the two anomeric carbon of one sugar and the oxygen atom of the other. It is a O-glycosidic bond because oxygen is involved in this process. So glycosidic linkages are named from left to right, mentioning the carbon numbers and the alpha beta configuration. Example, maltose. Maltose is the condensation between two glucose unit. The two glucose molecules are linked by alpha 1,4 glycosidic bond. This means alpha anomeric carbon or C1 of one glucose joins the hydroxyl group on C4 of the next glucose. Maltose is a reducing sugar, so it has free anomeric carbon. Digestible disaccharides in food, sucrose, lactose and maltose. Sucrose is the bond formation occurs between glucose and fructose. In case of lactose, the bond formation occurs between galactose and glucose, and in case of maltose the bond is formed between two glucose molecules. This is the structures of the digestible disaccharides in our food, like this is sucrose, this is lactose and this is maltose. In glycoproteins, we know two main types of glycose oligosaccharide linkages are exist. O-linked oligosaccharides attach to the protein via O-glycosidic bonds to the OH group of serine or threonine side chains. N-linked oligosaccharides attach to the protein via N-linked glycosidic bonds to the amino group of the amino acid like asparagine side chains. Important disaccharides like lactose, which is known as milk sugar composition, it is made up of galactose and glucose. The linkage is present in lactose is the beta 1,4 glycosidic bond. The chemical name of lactose is O. O means O-linked glycosidic bond is here. O beta D galactopyranosyl 1,4 D glucopyranose. Here the glycosidic bond, O-glycosidic bond is formed between a glucose molecule and a galactose molecule. It has a free anomeric carbon, hence lactose is a reducing sugar. It is found naturally in milk 0 to 7% depending on the species. What is lactose intolerance? Enzyme beta D galactosidase or lactase basically hydrolyzes lactose to form glucose and galactose. But in case of adults, especially of African and Asian descent, lactase activity decreases, so lactose remains undigested and it can be fermented by gut bacteria. It produces carbon dioxide, hydrogen and acids, causing bloating and pain. So lactose free milk is available, lactose hydrolyzed enzymatically. Lactose intolerance. What is lactose intolerance? We know in infants, lactose, also known as milk sugar, is hydrolyzed by the intestinal enzyme beta D galactosidase or lactase to its component monosaccharides for absorption into the bloodstream. The galactose is enzymatically converted or epimerized to glucose, which is the primary metabolic fuel of many tissues. Since mammals are unlikely to encounter lactose after they have been weaned, most adult mammals have low levels of beta galactosidase. Are you lactose intolerant? What happens consequently, much of the lactose they might ingest moves through their digestive tract to the colon, where bacterial fermentation generates large quantities of carbon dioxide, hydrogen and irritating organic acids. These products cause the embarrassing and often painful digestive upset known as lactose intolerance. Lactose intolerance, which was once considered a metabolic disturbance, is actually the norm in adult humans, particularly those of African and Asian descent. Interestingly, however, beta galactosidase levels decrease only mildly with age in descendants of population that have historically relied on dairy products for nutrition throughout life. Modern food technology has come to the aid of milk lovers who develop lactose intolerance. Milk, in which the lactose has been hydrolyzed enzymatically, is widely available. Sucrose. The most abundant disaccharide is sucrose. We know the major form in which carbohydrates are transported in plants. Sucrose is familiar to us as common table sugar. The systematic name or chemical name for sucrose is O alpha D glucopyranosyl 1,2 beta D fructofuranoside, indicates that the anomeric carbon of each sugar C1 in glucose and the C2 in fructose participate in the glycosidic bond formation, and hence sucrose is not a reducing sugar. Non-carbohydrate molecules that mimic the taste of sucrose are used as sweetening agents in food and beverages. Similarly, in sucrose, anomeric carbon atom of a glucose unit and a fructose unit are joined through alpha 1 to beta glycosidic linkage, and the configuration of this glycosidic linkage is alpha for glucose and beta for fructose. So the abbreviated name of sucrose is glucose alpha 1 2 beta fructose. Invert sugar. When sucrose is hydrolyzed, it gives glucose and fructose in 1:1 ratio. This mixture is basically called invert sugar or invertose because the optical rotation changes from dextrorotatory sucrose to lever rotatory fructose. Uses in confectionery, jams and syrup preparation. It's sweeter, it resists crystallization and retain moisture, and it improves texture. Trehalose. Trehalose is found in insect hemolymph, yeast and fungi. It serves as an energy source and protective sugar against stress. Maltose. It is produced during starch digestion by amylose. It is found in germinating cereals. The linkage found in maltose is alpha 1,4 between two glucose units. This is the structure of trehalose. This is the unit, and this is the units of maltose. How maltose is formed. Polysaccharides or glycans. Polysaccharides are the ubiquitous in nature and also called as glycan. On the basis of their functions, glycans are of two types: structural glycan and storage glycan. Structural glycans include cellulose, hemicellulose, lignin, suberin, chitin, peptidoglycan, etc. They provide mechanical strength to cells, organs and organisms. Storage glycans basically supply energy such as starch and glycogen. We know polysaccharides are the long chain of monosaccharides linked by glycosidic bonds, and so glycogen, which is an example of storage glycan, it can be homopolysaccharide or heteropolysaccharide glycan. Homopolysaccharide means contains only a single type of monomeric unit, and heteroglycans or heteropolysaccharide include contain two or more different kinds of monomeric unit. This is the examples table showing the two different types of polysaccharides: homopolysaccharide and heteropolysaccharides. Homopolysaccharides or homoglycans, they contain only one type of monosaccharide. We have already discussed. The first one is the starch found in plants. It is the storage form of glucose. two components it has, like amylose and amylopectin. Amylose is a linear chain structure. It has alpha 1,4 bonds, and amylopectin is a branched chain. Alpha 1,4 main and alpha 1,6 branches are present in every 25 to 30 glucose residues. Iodine test, it forms a deep blue or black color due to complex formation with amylose helices. Starch is formed by the formation of glycosidic bond in between glucose molecules. Now, the storage carbohydrate, storage polysaccharide in animals is the glycogen. It's a highly branched polymer of D-glucose. It has chains have glycosidic linkages. It's a storage form of glucose found in animal liver and muscle cells. It is highly branched like amylopectin, but branches occur every 8 to 10 glucose units. Bonds are here. Present is alpha 1,4, which is found in the main chain, and alpha 1,6 is present in branch chain. Cellulose is the structural carbohydrate or polysaccharide found in plants. There is a linker. Cellulose is a linear unbranched homopolysaccharide of D-glucose units joined by beta 1,4 glycosidic linkage. It is a structural polysaccharide of plant cells, although cellulose forms a part of the human diet. Such as in vegetables and fruits, it is not hydrolyzed by human enzyme system. So cellulose is one of the most abundant organic compounds in biosphere. We know human cannot digest cellulose. This is the structure of cellulose. Chitin is a linear homopolysaccharide composed of, it's a polymer of N-acetyl D-glucosamine, which glucosamine residues jointed by beta 1,4 glycosidic linkages. The only chemical difference from cellulose and in chitin is the replacement of the hydroxyl group at C2 with an acetylated amino group. It is a structural polysaccharide found in the exoskeleton of insects, crustaceans, and found also in the cell wall of fungus. It provides hardness and protection in the exoskeleton of the insects, crustaceans, and fungus. Inulin, it is a polymer of D-fructose units. The bond found in it is the beta 2,6 linkage. It is found in onion and garlic bulbs. It is used for glomerular filtration rate GFR in kidney function test. This is the structure of inulin. Foods high in inulin are the wheat or brown bread, salads and red onions, chicory oaks, rye, the leeks, etc. This is the structure of callose. Callose is the polymer of D-glucose, and the bond present in the callose is the beta 1,3 linkage. It is found in the plant cell plate formation, which is formed during the cell wall formation and pollen tube development. You have already studied about it. Now the last one is the dextran. It is found in dental plaques. It is produced by some gram-positive coccus bacteria. This is the structure of dextran. The main linkage found in dextran is the alpha 1,6 with branches at alpha 1,3, and sometimes it can be alpha 1,2 or 1,4. Now we should study for the heteropolysaccharides. It is also known as heteroglycan. It contains two or more kinds of monosaccharides such as glycosaminoglycans or GAGs, or it is also known as mucopolysaccharides. It is basically found in connective tissue. It is made up of repeating disaccharide units of an acidic sugar like glucuronic acid and amino sugar like N-acetyl glucosamine and N-acetyl galactosamine. It is negatively charged and unbranched. The GAGs are following hyaluronic acid. Its composition is glucuronic acid and N-acetyl glucosamine. It is found in synovial fluid and in vitreous humor of eye. Its main function is the lubrication. Chondroitin sulfate, it is made up of glucuronic acid and N-acetyl galactosamine. It is found in cartilage and bone. It provides a structural support. Keratan sulfate, the composition of what is galactose and N-acetyl glucosamine. It is found in cornea and cartilage. It acts as shock absorber. Heparin, it is made up of ioduronic sulfate and N-sulfo D-glucosamine 6 sulfate. It is found in liver and muscles. It acts as an anticoagulant. Heparan sulfate, it is similar to heparin. It is located at the basement membrane and its function is cell surface function. Darmatan sulfate, it is made up of L-iodurate and N-acetyl galactosamine 4 sulfate. It is found in skin and blood vessel. It provides structural integrity. These are the structures of glycosaminoglycan, chondroitin sulfate, darmatan sulfate, keratan sulfate, heparin, hyaluronic acid, peptidoglycan, agar, etc. So, in today's lecture, we explored the biochemistry of disaccharides and polysaccharides, understood their structure, types, and biological importance, and so how these complex carbohydrates act as both energy sources and structural components in living organisms. From sucrose and lactose to starch, glycogen, and cellulose, each plays a vital role in sustaining life. Remember, these molecules are not just sugar, they are energy backbone and structural framework of all cells. If this video helped you, don't forget to like, share and subscribe for more clear and exam oriented biology lessons. Thank you.